My screen is visible.
Yes.
Okay. So today we are going to discuss about the further topics and the questions related to that. So this is your week 5 assignment. Let's start this. Am I audible? Like if there is any problem, please let me know. Okay, let's start the session. In this week you have studied about different downstream processing methods and different type of techniques. So today all the questions are related to that only. Here is your first question. Let's read it. What they are saying: which sterilization method follows the time temperature relationship as stated below? So this graph or this figure is showing the temperature and time relationship, and this is mainly focusing on the sterilization method, or this is a graph of the sterilization method. So you have to tell which sterilization method this type of temperature and time relationship occur. So let's read the options. First is direct steam injection sterilizer. Second one is plate heat exchange sterilizer. Third one is indirect heat exchange profile, and the D option is none of the above. So this figure is representing which type of sterilization? Okay, first we must know what is happening in this graph. So we have seen that here this temperature is 27, and this temperature they have shown as 27 degrees Celsius, and when there is finally they are showing it as 144 degrees Celsius, means at this temperature sterilization is occurring. So what is happening? The temperature is rising up to 144 after 20 seconds. Means initially a little time gap is given. Then 2 to 3 minute sterilization of the component or any media, whatever you are sterilizing, that is occurring. And then again after 20 seconds, the temperature falls below. So this type of sterilization method is example of continuous sterilization. In continuous sterilization, you have two different types. First one is direct steam, and second one you have plate steam sterilization method. So this graph is representing the plate steam sterilization. Means in this initially there is a time gap, even that is very small, but initially a little bit of time is given. After that, that temperature of 144 is occurring. In direct stream, what happened? When you draw the temperature and time graph, the initially it has this high temperature, that is 144, suppose in this given. So 144 degrees Celsius direct this temperature is given, and after that 2-3 minute of time it will fall down. So this is the example of direct steam. In this, directly the solution or the inoculum is sterilized at 144 degrees Celsius, but in this a time gap is given. These both are examples of continuous sterilization method. So for this question, answer is plate steam, plate heat exchange sterilization method. Okay, yeah. Now coming to the second question. This is very simple question and very important question, because in this you will know like stepwise separation process. So you must know what you are doing at every step. So they are asking the following are the steps considered for a separation process. Arrange them in correct order. Means you have to tell which process you have to do first and then later on. So they have given first one is pre-treatment, second is solid concentration, C option is C is solid separation, and D is post-treatment. So you have to arrange them in a sequential order. So you know from the that initially that you have done is pre-treatment, means you are initiating the separation process. So there are some few methods that you have to do. Just like if you have collection of stones, so you will separate the stone on the basis of their size. So like initial treatment that you have done either manually, or you can also use chemical or physical method, but they are like very basic method, like you are changing the pH so that some things will separate, some components will separate down. So in this way, so initially you have done the pre-treatment, and post treatment is done at the later stage. After final separation you will do this post treatment. So the that starting from the pre-treatment you have to go till post-treatment. In pre-treatment there are different methods people are using, but they are very basic method, like different some chemicals they can use, some physical methods are also used. Then after what happened after pre-treatment, you initially concentrate the solid part. So this solid concentration will occur, and after this solid concentration, like suppose this is your solution and you have different sizes particles. So what you will do, because you can, so first you try to like settle down or collect down all the solid material at a particular place, then you will do the separation. So after that you will separate that solid from the liquid part. So this is the order of the separation process. In this order they have given also that first one is the pre-treatment, second one is solid concentration, third one is solid separation, and the last one is post treatment. For the solid separation you also have different method. For solid concentration you also use different method, centrifugation, precipitation. For solid separation you can use filtration and other methods also. Okay, but this is the basic process that can apply to any type of separation, for separating any component from pharma industry, from like alcohol also you can use. This is the like basic steps that are to be done for a separation of any type of particular. Okay, so for this question answer is A, B, C, D. So that option A is correct. Now let's read the third question. Which of the following mechanism is followed during filtration? So they are asking about the filtration mechanism. All these things you have studied in your last session. So there are four different type of four mechanism for this filtration. They are okay. They are given here. First one is straining, second one is impingement, third one is entanglement, and fourth one is attractive forces. These are the different mechanism, like how the filtration is occurring. Suppose in a straining, what is happening? Straining is very simple. Of first time you can say at a very basic level that filtration is occurring on the basis of size that we use in houses also. So it's like a sieving. Straining is the simple filtration. The larger particle will stay there and the other particles will flow down or the liquid membrane separated. In impingement also the separation is occurring on the basis of the size. In entanglement there is also separation on the basis of size. But what happen in the entanglement, suppose this is your filter paper. So in this filter paper what happen, the there are different size of pores in this filter paper. Suppose so the particles will entangled or get get immobilized, not immobilized but get attached in the spores of this filters. Understood? This is the entanglement, means the particle are settling in between the pores that are present in the filter. In between the filters, the pore sizes, in the pore sizes the molecules are settling down. So this is the type of, this is third type of mechanism for filtration. And the fourth one is attractive forces, means if there is any type of electrostatic force is present between the filter and the particles that you want to separate, then this type of attractive force is occurring and the filtration occur. Okay, these four different type of filtration mechanism are there. So okay, and further filtration is also can be divided into two types, that on the surface and depth filtration. In what happen in surface filtration, suppose this is your, sorry, this is your filter. So the separate or that solid particle will settle above this, like on the surface of the filter paper. The particle or solid particle get collected here on the surface of this. And in the depth filtration what happen, suppose this is your filter paper. I'm just trying for your visualization in a 3D type of. Okay, suppose this is your filter paper. So what happen, that solid particle will settle here in the space between the pores that present in the filter. So this is the type of depth filtration and this one is type of surface filtration. Okay, and these are the mechanism. So in these mechanisms can be divided into both the depth filtration and surface filtration. Okay, understood this filtration thing? Because this is really important and given in great detail for your syllabus. So you must know every aspect of this filtration. So if anyone has any question, please ask. Okay, so everyone understood this. So let's mark the answer for this question. First one they have given that entanglement. So yeah, right. This is also a mechanism of filtration. Second one is straining. This is also a mechanism of filtration. So C, both A and B. So for this question answer is C. Now coming to the fourth question. This is also related to filtration. Which of the following equation are valid for the filtration process? Means if anything is filtering out, they are talking about the rate. So let's read. First option is rate of flow. Rate of flow is equal to driving force upon resistance. Normally if something is filtering out, suppose this filter paper you are using, so and the a liquid part is filtered out and the solid part is separated or collected on the filter paper. Then this rate of flow is equal to, why this flow is occurring? This flow is occurring due to presence of pressure, dif- or you can say force. So this difference and the resistance is also occurring. So this rate of flow usually we define as driving force upon resistance. So this one is correct, that rate of flow is equal to driving force by resistance. Now let's read the second option. In second option what they are saying, that rate of filtration is equal to dV by dt. Means in second option what they are saying, that rate is equal to dV. What this dV is representing? dV is representing the change in volume with respect to time. Wherever you get this d, that differentiation d is mainly representing the change. Okay. So rate of filtration can be defined in both way, like this is dV by dt also, and rate of flow is equal to driving force by resistance. That is also true. So for this question, answer C, both A and B. Both these reactions can be used to represent the filtration process. Okay. Either you can represent the rate of flow in the form of driving force by resistance, or you can represent in the change of dV by dt, that is rate in change of volume. So for this question, answer is C. Anyone has any doubt till here? Okay. This is our next question. In the chromatography technique, which solvent carries the analyte? So options are eluent, eluate, both A and B, and D none of the above. So what chromatography is? Chromatography is also a separation technique, because in this what happened, the separation is based on the solubility in two different phases, that is stationary phase and mobile phase. From the name it is clear that this is a stationary, means this will like stay at a place, it will not move, and the mobile phase, the solvent that is moving. Okay, suppose, just a minute, if we are, this is column, usually we use column in all the different types of chromatography. This column will vary from the type of chromatography, but usually the separation is occurring in this type of the column. So a stationary phase is that is present inside the column, or on which depend the separation is occurring. Suppose these beads are present there. Okay, I will explain you. These are the stationary phase. And the solution, like you want to separate any protein which is present in the solution. So this will work as a mobile phase, that you will load in this column and finally the separation will occur. That separation is based on different types. Suppose if you have like you have example ion exchange chromatography, you have size exclusion. Third one, suppose you have affinity chromatography. So what is happening in all the different type of technique, this stationary phase will differ and according to which separation is occurring. And this, how do we choose which type of chromatography is required? That also depends on what you want to separate. Suppose if you have a mixture of, suppose you have a mixture of three different molecule, one is bigger size, one is small, and one is very small. Means you know they all the three component that you want to separate are of different sizes. So what you will do, you will try to separate them on the basis of size. Means you will choose the size exclusion chromatography. In size exclusion, what will happen, you will have the beads of different type, and the separation is occurring either the s- this solute is getting into the beads, it will pass either through the beads or either outside the beads. So it will depend on the size. Okay, so this is the mobile phase that we need to that need to be passed or allowed through the stationary phase, and finally you will collect your sample here. So the sample that you get collected here, it is called as eluate. Okay, but the solvent through which your analyte is passed is called as eluent. So you need to learn, or you need to know all the terms that what is eluent, what is eluate, what is stationary phase, mobile phase, and how you choose these different types of chromatography. Although you don't need to go in very detail, but very basic you need to know all those things, what are these, and especially these terms, eluate, eluent, stationary phase, what is mobile phase, how they will differ. These four terms you must know about the chromatography. Okay, so now come to the question that in the chromatography technique, which solvent carries the analyte? First one is eluent. So yeah, in eluent your analyte is present. Analyte is referring to what you want to separate. Okay, so this is present in eluent. Yeah. So for this, correct answer is A. Now coming to the next question. What is the material for plate and frame filter press? So anyone want to tell where you use this plate and frame filter press, or what is this? Okay, you all have studied about the filtration and different type of filters. So this is, you have studied different types of filter. This is one type of filter. Okay. So what is this usually made up of? They are asking this. First one is stainless steel. Second one is aluminum. Third is galvanized iron. And the D option is borosilicate glass. So this type of filter press, these type of filters are mainly made up of aluminum. So correct answer to this question is B. Okay, because we cannot use iron because that will corrosion will occur, that can break. And stainless steel also we don't use because that will not like can take that much force you will require. So for this usually aluminum is preferred. The next question is the example of membrane filtration process is or are. So they are asking the membrane filtration. First one is ultrafiltration, second one is reverse osmosis, C option is both A and B, and D none of the above. You will know very easily from the question that they are asking that type of filtration you need to tell in which you are using the membrane. Like different type of membrane occur. Suppose semi permeable membrane you usually use for the filtration. Okay, someone answered about this question. Okay, let me see. Answer A. So for this question number 7, you are answering this. Atendra Karar.
Yes, ma'am.
Okay. But reverse osmosis we also use filter na. We also use membrane in reverse osmosis process. Ultrafiltration is also a type of membrane filtration, but reverse osmosis is also a type of membrane filtration. So for this question, answer is C. Yeah, right. Sadvika. You all, can anybody want to tell about the difference between osmosis and reverse osmosis? Like, although it is very clear from the name itself.
In osmosis, it will be from higher to lower, but in reverse osmosis, it will be reverse process, means lower to higher concentration. The flow will occur.
Yeah, right. It's concentration reverse. So let's. So for this question, answer is C, and I will tell you like how these membranes are usually selected. This is for osmosis and reverse osmosis. Because normally why in any movement will occur if there is difference in level, means either if there is something at a higher temperature and another thing that is at lower temperature. Sorry, not temperature, if it's anything higher concentration, temperature, pressure, normally the movement will occur when there is a difference. So in this osmosis and reverse osmosis, in in osmosis that is occur on a normal pressure, but in this reverse osmosis you have to apply pressure from the outside, and after that the separation will occur. Okay, for this answer is C. But how this ultrafiltration and reverse osmosis, even you have studied about the microfiltration, macrofiltration, so how these filtration membrane will occur, or how to choose the filtration, which filtration is required for which process, or the separation of what molecule or what component. So this is really important that all these are basis on the basis of size, molecular weight also, but the main factor here is size. That the very small molecule, or as the size decreases, you need a filter membrane or filter that can separate the very minute particle. So according due to so on the basis of this there are different type of separation processes occurring. Like if you want to separate the larger molecule, you can use directly filtration, or you can call this particle filtration. Then you will go to microfiltration when the size of the molecule will go down in the micro range. Then ultrafiltration when you will require for the very small particles. And then nanofiltration, nano itself like defined for the -9 or nanometer range. Then reverse osmosis. In reverse osmosis, very rarely minute particles will allowed, otherwise it will not allow the particles to pass through this. So all these things need to be done on the basis of size only. Okay, so this is really important that you should know the order of this also, at which filter will used at what order. Okay, yeah, next question. That non-volatile components in the fermentation medium can be separated by. There are four options. First one is crystallization, second one is evaporation, third one is distillation, and the fourth option is freeze drying. So they are asking about the non-volatile component. So what do you understand by the non-volatile component? Anyone? Okay. Okay, non-volatile component are those which are not directly convert or can be converted into gaseous form. Okay, so these are the non-volatile compounds which cannot be like converted or go into their gaseous form. So you need a fermentation medium that can be separate. Yeah. Okay, separation is for this molecule. Which method you will use? So let's discuss about all these four techniques. In crys- okay, let's start from the D option. In freeze drying also, what will happen? Suppose you have a this mixture and the if there is any liquid present that will go into gaseous form. Okay, in distillation what will happen? In distillation also the liquid is converted into gaseous form. In evaporation what will happen? Of in this also gas form is formed at the end. In crystallization, gaseous form is not required. So for this, because this is not converted into gaseous form, so these three techniques can't be used for this separation. So we will use the crystallization method because in this that conversion is not required. Okay, what will happen in crystallization? Mainly the suppose if you have a mixture like solid or liquid is present there, then that aggregation will occur, and finally according to their lattice the crystal will form. What is representing here lattice? The arrangement that you might have studied in your chemistry in 11th 12th level. So this that we define that phase atom, phase cubic or cubic, all these things, these define the this lattice of the component that finally form in a crystal form. Okay, so for this non-volatile component, you will use the crystallization method. Yeah, this is the next question. Which of the following statements about evaporators are correct? First one is industrial evaporators have mostly tubular surface. That's true. Then the fluid is circulated through the tubes. The tubes are heated by steam that increases the temperature of the outer surface. And the D option is all of the above. So please answer because all of you know this answer. Tell me how the industrial evaporators will work. Anyone please answer this? All these statements are directly written on your slides. And if you have seen lectures, you will know the answer. Okay. D. Yeah, right. This all the three statements about this evaporators are correct. That in industrial mostly we use the tubular or actually I haven't put the picture, but that picture of this is also present in your slide. That industrial evaporators are usually present in this form and the fluid will flow in a circular form, and it is also that if you speed up or how fast the fluid will occur, that is much better for this. this process. So if we speed up this circulation uh, of the fluid, then it means the better evaporation will occur. So for this question, all the things are correct. Means tubes are also heated by the steam. That's why their outer surface is also heated up. So the temperature of the outer surface is also increased. So for this, answer is D. This I have already told you that crystallization process involve which of the following separation is occurring in crystallization, that I have explained in last question only. Okay, crystallization process involve the following: solid solid separation, solid gas separation, solid liquid separation, and liquid liquid separation. So this method is mainly used for solid liquid separation. Okay, that I have discussed in the previous question. So for this, answer is C. Someone answered B. Then I have explained in this question we discussed no that crystallization cannot be used, can be used for non-volatile component. So this can't separate the solid gas. Because if, or you can understand in that way that if you have that mixture, like in this crystals are forming, but this is mainly used for the mixture of the solid and liquid separation, not for the solid gas separation. Okay, this you all must know that which of the following alcohol is are not toxic for human consumption. So what are the options? First one is Tex alcohol, second one is non-Tex alcohol, third one is rectified spirit, and the D is all of the above. A, yeah, right answer. Tex alcohol, this is the alcohol that is used for consumption. Okay, this question: which of the following extracellular enzyme is responsible for the conversion of starchy material into soluble sugars? So you have all these four options are given the different enzymes. And in the first lecture I have discussed that in this course the enzymes or the use of enzymes is really important. Yeah, Satvika, correct answer. So let's discuss but how all these four enzymes are used for the conversion. First one, we have already discussed in previous assignments that cellulase is used for the degradation of cellulose to reducing sugar. So this is not used for starchy material. This molasses enzyme is mainly used in sugarcane industry. This molasses is used in sugarcane industry. This lipase is also used for the reduction of fat. And this amylase we used for this conversion of starch molecule into soluble sugar. So this amylase is used for starch conversion. So for this, answer is C, but you must know all the four enzymes or even other enzymes also that you can find in different books. So you must know the function of these enzymes, like few major enzymes. Not all the enzymes you can learn, but the major enzyme you must know. So for this, answer is C. Okay, which of the following metabolic pathway is followed during ethanol fermentation? So this is also very easy question. First one option they have given von Liebig and Pasteur theory. Second one is EMP pathway. third one is bifidum pathway, and D is none of the above. Okay, yeah, Satvika, correct. For this ethanol fermentation, what pathway is used? This is EMP pathway, that is mainly used for ethanol fermentation. These things you will know directly if you have even seen the lectures once only, or if you have gone through the slides. Even normally, if you have studied this ethanol fermentation in any way, you have studied this EMP pathway. Anyone want to tell me the another name of this EMP pathway? Yeah, glycolysis. Yeah, correct. So this is also called as glycolysis. So for this question, answer is B. Okay. Let's read the question. A fluid flows through a pipe of diameter 0.2 meter under a velocity of 0.8 meter per second, and if the Reynolds number for the system is 2600, what will be the kinematic viscosity of the fluid? So they are asking about the kinematic viscosity. They have given kinematic viscosity is defined as the ratio of dynamic viscosity to the density of the substance. They have provided you the definition. So for doing this, what they have given, that Re, Reynolds number, is usually represented with Re, that is 2600, and they have given the diameter of the pipe, that is 0.2 meter, and velocity v 0.8 meter per second. And what they are asking, this kinematic viscosity, that is usually represented with like mu. Okay, so we need a equation that will like represent the relationship between this Reynolds number and the kinematic viscosity. So we have direct formula for this, that this kinematic viscosity is equal to that v, this v is representing here velocity, into d, that is diameter, and upon Re. Or you can write it reverse, that Re, Reynolds number, is equal to v into d by this kinematic viscosity. So this is the formula that you all will know directly by putting the values. We have this v, we have d is also there, and Re is also given. You only need to find out this kinematic viscosity on you. So you can solve it directly from here, that is 0.2 to 0.8 by 2600. So tell me the answer by solving this, because only calculation you have to do, and that is also very simple. Tell me what will come. Okay, B, 6.15 10 raised to the power minus 5. Yeah, correct. When you solve this, it will come this. Others, yeah, Satvika, yeah, correct. If you write it, it will come like 6.15 into 10 raised to power minus 5. So correct answer for this question is B. Now this is the last question for today's class. What they have given, that ethanol fermentation using glucose as a substrate follows the following reaction. That is, sugar is converted to alcohol plus carbon dioxide plus cell mass if formed there. This is a like general equation of the ethanol fermentation. Any type of sugar you will take, these type of components or combination will form. Now what they have given, they're given that from the balanced equation it was found. So balanced equation means that stoichiometrically balanced equation. Okay. It was found that Y ethanol by substrate is 0.51 gram per gram. What will be the value of Y CO2? Means this Y is representing here yield. Okay. If no cell mass formation occur, means this sugar is only converted to that alcohol plus CO2. And in this they have also given specifically that is ethanol. So and there is no cell mass formation, means there is no other product is forming. Means sugar is directly converted to ethanol and CO2. And from the balanced equation they have found that yield for the formation of this is 0.51 gram per gram. Okay. Then how much CO2 will form? Suppose this is 1 gram sugar is there. So from 1 gram, how much alcohol is forming? 0.51 gram. So how much CO2 will form? The remaining part of the sugar that will form the CO2. So it will become like 1 minus 0.51. So how much it will come? It will be 0.49. That is gram per gram. This is very simple question. Like if you suppose that 100 parts are or 100% sugar is there, then how much alcohol is forming? 51%. Because they have given yield 0.51. So how much CO2 will form? The remaining part of the sugar, if 51 is used for the formation of alcohol, the remaining part will be used for the formation of CO2 because there is no other product. And they have also given that the equation is balanced, so you don't need to think of that. You can directly use this, that if one is this sugar and 0.51 alcohol is formed, then the remaining part will form the CO2. So for this question answer is B. Okay. So answer to this question is B. And yeah, this is the last question for today's session. So if you have any doubt, then please let me know. We can discuss the question again. No, ma'am. Okay, Satvika, last question. Yeah, we'll discuss again this one. Just a minute. Yeah, they have given that ethanol fermentation using glucose as a substrate follows the following reaction. Means if you see any ethanol fermentation, this is the general reaction that is occur. So from the balanced equation, when you balance this equation, it was found that Y means yield, means how much is forming. They have given a Y ethanol upon substrate is 0.51 gram per gram. Or you can understand this, that ethanol, what is ethanol, is the alcohol that is forming. And if you consider substrate as sugar, so this is they have given, if you consider this, that from 1 gram sugar 0.51 gram of alcohol is forming. So this is the meaning of this Y, or yield of ethanol by substrate 0.51. That from 1 gram substrate of sugar, this 0.51 gram of ethanol or the alcohol is forming. Then they are asking what will be the value of Y CO2 by substrate. Means they are asking how much CO2 will form then, or what is the yield especially. If no cell mass formation is occur, means there is no cell mass is 0. So it means from 1 gram, 0.51 gram alcohol is formed. Then the remaining part of this substrate, what it will form? It will form the CO2 because there is no third product is forming. So from 1 gram, how much is remaining for this CO2 production? 1 minus 0.51, because this is already used for alcohol formation. So it will come around 0.49. So it means that 0.49 gram of CO2 is forming from 1 gram of this sugar part. That 1 gram sugar, 0.51 is gone for the alcohol formation and 0.49 is gone for the CO2 formation. And only these two products are forming. Okay, now understood, Satvika?
Yes, ma'am.
Okay. Any other doubt?
Ma'am, that 1 gram is we are taking it as a standard value, or we should take it as only 1 or 100 also we can take, ma'am.
You can also take in the form of 100. Like you will consider this as a percent, because the option or the value that you have to answer, usually yield we provide in the form of gram per gram. You can do it by using 100 also, because from 100 if you will take, like suppose from 100 you will consider this 51 will form the alcohol, then the remaining 49 will be forming this CO2. But when you will answer, that is 49 upon 100, so it will come 0.49, that is gram per gram. So you can take any value, because whatever your substrate, you are telling it in the form of yield, so that will not change if you take the value as 110 or whatever.
So the total value we can take it as 100 or 1, right, ma'am?
Yeah,
We
right.
can take anything.
You can take anything.
Yeah, okay.
Okay. So now, okay,
Thank you so much, ma'am.
welcome. Let me close the recording.